Hello, everybody aspirants. This is a Unit 6 review of the chapter Gene Expression and Regulation. So DNA, which is deoxyribonucleic acid, it is also called blueprint of life. It is a double-stranded, as you can see from the picture, that contains the genetic instructions used in the development and function of all organisms. DNA is made up of nucleotides. Each nucleotide has a deoxyribose sugar in case of DNA and ribose sugar in case of RNA, phosphate group, and nitrogenous bases. Nitrogenous bases are of two types, purines and pyrimidines. Purines have adenine and guanine, and pyrimidine have cytosine and thymine, which means that DNA has 4 NTPs, DATP, DGTP, DCTP, and DTTP. DNA and RNA both are the primary source of heritable information because they contain the genetic code that is passed down from one generation to another. The first process which we have to learn in this chapter is DNA replication. So it is the process by which cells copy their genetic material before cell division. DNA has two strands, a leading strand which copies from 5' to 3', and lagging strand which has 3' to 5'. Both strands run anti-parallel to each other. Now, during DNA replication, we have just one DNA polymerase to take care of both the strands for making copies of DNA. To begin with DNA replication, the enzyme called helicase and gyrase help in opening up the DNA so that it forms a replication fork like this. Now, both the strands are exposed and DNA replication starts with the help of DNA polymerase enzyme. Since DNA polymerase moves in only one direction, that is 5' to 3', NTPs which are added in the lagging strand, they are added in a slow manner. Whereas in leading strand, the DNTPs are added at once. The small fragments which are formed in lagging strand are called Okazaki fragments. And later on, these fragments are sealed with the help of ligase enzyme. And thus, copy of DNA is made. The double helix structure of DNA allows for easy replication through a semi-conservative mechanism where each strand serves as a template for the synthesis of a new complementary strand. So replication requires unwinding with the help of the enzymes helicase, as I mentioned, complementary base pairing with the help of the enzyme DNA polymerase, and joining, which is done with the help of the enzyme ligase. And DNA polymerase also helps in sealing the gaps. This is the semi-conservative model, which shows us that both the parents, when they recombine, the offsprings have parental as well as hybrid copies. Parental copies as well as recombinant copies. The next process is transcription, where DNA converts to RNA. Since mRNA, messenger RNA, is a single-stranded molecule, so only one strand of DNA copies. Only one strand of DNA forms complementary base pair. Because RNA is also 5' to 3', and DNA has two strands, one is 5' to 3', another is 3' to 5'. So the 3' to 5' strand copies makes complementary base pairs for RNA. DNA is transcribed to messenger RNA. The sequence of bases in the mRNA molecule codes for the specific amino acid sequence of a protein. So once mRNA is formed from the 3' to 5' strand of DNA, the mRNA sequence has codes, 3 letter codes like this, which help in formation of protein. The triplet in mRNA sequence are called genetic code or codon. So AG codes for amino acid number 1, and then next triplet codes for amino acid number 2, and so on. tRNA or transfer RNA has specific binding sites for specific amino acids as well as an anticodon sequence that base pairs with the codon sequence of the mRNA. So the job of tRNA is to help mRNA and proteins communicate with each other, because mRNA is made up of nucleotide bases and proteins are made up of amino acids. So we need a code. tRNA has one anticodon arm which sits on the mRNA and one amino acid arm. This is the structure of the ribosome on which mRNA sits. After transcription, once mRNA is formed, the next step is RNA processing. RNA processing includes 3 steps. Addition of a poly-A tail, also called polyadenylation. It helps in protection of mRNA from degradation and also helps in transport of mRNA out of the nucleus. Second step is addition of 5' cap. The cap helps to protect the mRNA from degradation and also helps in recognition of the mRNA by the translation machinery. Next is removal of introns. The introns are removed by a process called RNA splicing, and this process is catalyzed by a complex of enzymes called the spliceosomes. The exons are then joined together to form a mature mRNA molecule that contains only the coding region. There is also one more mechanism in splicing, also called alternative splicing. Alternative splicing is one of the reasons why number of proteins are more than number of DNA sequences. A major mechanism of gene regulation in eukaryotic cells, and it plays a key role in the complexity and diversity of eukaryotic organisms. So this is a mRNA which is processed with a 5' cap, 3' polyadenylation, and later on the introns. If you can see these lighter regions, these are the introns regions which are removed, and only exons, that are the coding regions, are joined together. This mRNA is prepared for translation. The sequences which are present in the mRNA will form a 3 letter codon, and each codon will code for an amino acid. For example, AAA codes for lysine, AAU codes for asparagine, AAG also codes for lysine, and so on. Next is translation. Translation means conversion of mRNA to polypeptide chain. On the ribosome, this mRNA sequence is bound. The last subunit of the ribosome contains 3 pockets, K, P, and E. tRNA, containing amino acid arm, sits and helps in combining one amino acid with another. Translation is a process by which genetic information encoded in mRNA is used to synthesize a polypeptide, a chain of amino acids that form a protein. The first step is initiation. For initiation to begin, rRNA, that is part of ribosome, interacts with mRNA at the start codon. Next step is elongation. The tRNA in the A site brings in the next amino acid, and the ribosome catalyzes the formation of a peptide bond between the amino acid in the P site and the incoming amino acid in the A site. Third is termination. Recognition of one of the 3 stop codons leads to termination. So if any of these 3 stop codons are encountered, the polypeptide synthesis stops. Gene expression can be regulated at different steps. So in prokaryotes, group of genes called operons are transcribed in a single mRNA. First is inducible operon, also called lac operon. In case of an inducible operon, the operon is normally shut off, and it works only when the inducer is present. Here the inducer is lactose. This lac operon basically deals with formation of or lactose metabolism. In normal circumstances, the structural genes are not working because RNA polymerase is not bound to the promoter region, and there is a repressor bound to the operator region. Because of the repressor bound to the operator region, the structural genes are not able to transcribe. When lactose is there in the cell, this lactose goes and binds to the repressor and makes it inactive. Once the repressor becomes inactive, the promoter can bind to the RNA polymerase enzyme, and structural genes can transcribe and form the enzyme beta-galactosidase. Next is tryptophan operon, also called repressible operon. This is called so because this operon normally keeps on working all the time. The function of this operon is to synthesize tryptophan. It is repressed only when tryptophan itself represses or acts as a co-repressor. So whenever the levels of tryptophan go very high, they start inhibiting their own synthesis by acting as a co-repressor. So in normal circumstances, RNA polymerase is always bound to the promoter, and thus these genes are always transcribing and forming tryptophan. When the levels of tryptophan are very high, it acts as a co-repressor and goes and binds to the repressor site. Repressor co-repressor complex becomes active. Now, once repressor is bound to the tryptophan, RNA polymerase is not able to bind to the promoter because operator region is occupied by the repressor, and thus RNA polymerase cannot move and reach the structural genes for transcription. Regulation in eukaryotes. DNA have regulatory sequences that play a crucial role in controlling gene expression. The genes which are present inside our body, we don't want them to working all the time. So that's why we have a control over them. We can turn them on and turn them off whenever they are required and not required respectively. These sequences are located near or within the promoter regions of genes, and they interact with specific regulatory proteins to either activate or repress the transcription process. Next is mutation. Mutation can be of two types. One is point mutation, which affects only a single base, and frameshift mutation is the one which affects the whole open reading frame. This is the normal sequence. The first type of mutation is a silent mutation. In case of silent mutation, as we know that there are multiple codes for a single amino acid. We have seen that in the genetic code chart also. So if instead of C it becomes T, and GCC is also coding for alanine and GCT is also coding for alanine, then still it will remain the same sequence. That's why this type of mutation is called silent mutation. Nonsense mutation means earlier it was coding for cysteine, and because of this small change, now it is not coding for any amino acid, or probably it is coding for a stop codon. Missense mutation means where because of one single base change, earlier it was coding for cysteine and now it is coding for arginine. Now coming to frameshift mutation, their mutations are bigger level. In these type of mutations, it is either insertion, deletion, substitution of any of the base sequences, which leads to change in the whole reading frame of the amino acid of the protein. That's all for the video. Thanks for watching.